I will talk about three things. First of all, I will do a blah blah introduction. Then I will continue and say a few words about a ring shuffling project we did with EU money in the past, and show some results from that. And in the last part, I will show a bit about a way how we use synergy to make these Lantibiotics active against gram-negatives. So what you see in the history of antibiotic use is that new drugs are found and new resistances appear. And this repeats and repeats and repeats. And therefore, I think we should keep looking also for new antibiotics. But it's true that it's also very important to look at mechanisms of action. And it's also important to look at other ways of preventing infections. But I think it's very a multifactorial thing. You can't just focus on one thing and not do the other. So I think it's good to keep on looking. And the natural diversity is very, very big. And the kind of structures you can find are very interesting, I think. And well, I think you should keep looking. So what we focus on is trying to also really look into the ones that are critical. I realize that nobody can read this slide. But it's mainly gram-negatives. That's a bit the message here. There's also some gram-positives like Staph aureus and Strep pneumo. But yeah, we also really try to look into Pseudomonas and Acinetobacter to find new drugs. So we heard quite some of these compounds passing already. Yeah, so you find them everywhere, and apparently we have to add an arrow here somewhere. Where's tetracycline? Here, so we have to also do an arrow here now. But I didn't know this before, so I will adapt the slide. But we are focused on Lantibiotics and also other modified peptides that we use in our research. So why are we interested in Lantibiotics? Well, first of all, they are already a good starting point. They're quite potent against gram-positive bacteria. They have a low MIC value. They are synthesized ribosomally, and this is very nice because this means you become very flexible in changing the substrate for the machinery, but also making new drugs you can do by genetics or making new potential Lantibiotics. So they are modified heavily after translation. They have these lanthionine structures. They have a history of safe use. There's quite a decent chance that you consumed a few of these molecules this morning when you had some cheese or maybe during the lunch. But also there's a low rate of resistance development. And I will go into more detail about it later. So looking at the whole scheme of trying to find new drugs, well, you start off with the diversity that you can find in nature, and nowadays the sequences that are in public databases, you can use these to find new molecules, new ribosomal molecules, but also non-ribosomal molecules that the synthetase complexes you can find in the genomes. You can use that then to produce them, to screen for activity, do structural characterization, and so until here is where we are active until the patenting part in the Kuipers lab. It's a lot of work, and especially if you want to do this for every compound, it takes a very long time. So that's why we looked into making this more high throughput. And during my PhD, I started doing this by just doing it one by one. But later on, we found a more efficient way. And this was, I will explain later a bit more. So the biosynthesis of nisin, as not all of you are from the prokaryotic field, It is quite complex. There's 11 genes involved, and it makes the active compound. The active compound is also auto-stimulatory, so it activates itself to produce even more of the compound, and there's an immunity part. And the nice thing is that the last step is to cleave off the leader, and that activates the antimicrobial properties, and therefore if you do reduction or so, you can leave out the last step and make not active variants. So the biosynthesis starts with a leader peptide that is so the full-length protein. It's modified by a dehydratase and a cyclase, and they work in a directional fashion, and they do this in turns. And the nice thing is that you end up then with the rings. This is then transported, and then the last step is the leader cleavage. So then, how does it work? And yeah, well, Evian is in the audience, but you have two mechanisms of action. So one is that you bind lipid 2 and you sequester this, and the other is the pore formation. And there's a nice model for the stoichiometry here, where you have the 4 to 8 stoichiometry. So what we do to generate new variants is we have now we have 3 systems to produce these compounds. We can do it in Lactococcus lactis, in E. coli, but also in Bacillus subtilis, thanks to the Rafts for Biotech project. And we then normally express the new substrate using using the fusion with the nisin leader. You have the enzymes, and you can produce and secrete new molecules. So what we did for this first part is that we look at the lantibiotic itself, trying to use a synthetic biology approach. So we say these are all modules, and we can say per module we could exchange them or use them. So you have a lipid 2 recognition domain that by itself is active. We have a membrane insertion domain, and you have a hinge region, which allows for flexibility. But the point is that as you have many of these lantibiotics in a similar class, what you can do is you can say, okay, let's make new combinations based on what is available in nature, but yeah, let's make new variants. So what we did is per module is select different known lantibiotics, or even add synthetic parts, and then a library was synthesized containing all these substrates. So you can get a lot of options, and one thing to take into account is that we limited the first module because we want to be able to cleave the protein in the end with the original protease, the nisin P. So if we can't do that, then we can't activate. So therefore, in this setup, we limited ourselves to these 2 modules in the first part. So what we then did in a collaboration in the EU project, and the synthesis was done by the Wagner group, is all these variants were synthesized, and we got modular recombination, and we get all these plasmids. We transform them to a production host, and then we have to screen them. Now, how to screen them? This is a tricky part. For this, in the project, we had people from Sven Panke's lab, and what they have is a very nice system where you can use alginate beads to have essentially 3D mini reactors. And the advantage is that you can then have many, up to 25 million reactions in one grinder tube. So what we do is we have an alginate bead, and inside we incorporate one producer cell and normally many indicator cells. And the cells can grow to form a mini colony inside the alginate bead. So, and then also the bead is coated so that you don't get diffusion outside the bead. It's also important.
Okay, can I ask, how can you control the system? Can you, there is only one, one green thing and several...
Yeah, so that's a matter of statistics. So you add, you will see later in the results, so you add so little of the producer bacteria into your suspension that you expect 0 or maybe 1 in your droplet. That is important because if you have multiple producers, then yeah, it could be a combined effect and you don't know what you're screening for. So for this, they also have a COPAS setup, which is a particle sorting machine. Essentially, probably it's not fully correct, but it's like a fax. So you can select a certain fluorescence that you want, and then sort those out. And in this way, later on, you can then sequence what was active and find out what was the interesting compound. So what you see here is, or you don't see if you're too far in the back, but what you should see is you see many blobs, and those are like the beads, and you see that many of the beads have no red colony. In this case, the red colony is a producer. This is because we don't want 2 red colonies inside. And you see in this case, for instance, that not really an active compound was produced because you see the red and the many green cells are still active. And in this case, we see that also the red is a bit reduced. This often happens because, so the sensitive strain, in this case it's Lactococcus lactis, it's a very dangerous pathogen. Not really, but we are also using now different organisms, but for the setup here we were using lactis, and it's expressing the nisin protease, but the active compound also diffuses back to the producer, so you also see inhibition at the end of the producer cell of the producer colony. So what you then can do is so select, and what we did as a criterion is so it should have red fluorescence, but no, it should have no green fluorescence. That's the most important. And we took colonies that do have red fluorescence and colonies that don't have red fluorescence. Well, and you see we checked in this case, so this is the number of particles that we checked. So this is in the order of 100,000 or yeah, 100,000. And so of these, we only selected then in the end a very small amount. So then the next thing, and this is this has been quite some work, is to characterize. So I think we selected around 100 candidates, plus or minus a bit, and then the question is, okay, you see activity in this bead, but now what does it say? Because we want to improve from what we had as a starting point. And so what we did is and you can imagine that not only activity matters, but also diffusion capabilities and also modification degree, production, efficiency. So you're testing a lot of things actually at the same time. So what we did here is we corrected for the production level. And we did this by purifying a little bit on a larger scale and then cleaving it. And then we could use the leader as a indicator for the amount of protein. Because the leaders everywhere are the same. Of course, the peptide itself is different. So that's more difficult to compare. And then we check what the halo is on a diffusion assay. And then, so what you see here is that the red ones are interesting because they're the halo size, the relative halo size is 150% of, I think this is set to nisin, if I'm correct. Yeah? And in this way, we were able to screen find quite some new interesting variants. Oh yeah, this is something on the modular diversity. So we were wondering what do we find back, and we see that we find most that we found back have 3 organisms or 3 lantibiotics where the modules originate from. And you can see also that from the relative halo area, we found about, depending, so more than 125%, we found like 20 candidates that fall into that category. And here you can see a bit more in detail. So if there's a check mark, it means that we found that back in our selected candidates in the end. And then you see that Gelidermin and nisin are quite common to find back, and that especially in this module, the hinge region, you can find a lot of variety, but already also in module 3 we find quite some different. Here it's mainly nisin, but in the hinge region you see more diversity, and the same is also true for the 5th model, so the last part. So it's submitted and but not yet accepted. But these are the compounds. These are the best variants. It's not very detailed, but nothing I can do about that. So then we tested these variants, and what we find back is that we find improved variants, for instance, against Streptococcus pneumoniae. Here we can improve if you compare to Gelidermin or nisin. It's not a very shocking improvement, and it's also still gram-positive. But I said we would do something about gram-negative, so that's the next step. It's strange to me that lactis is not in here, because that's actually what we screened for, but I didn't make the slide. So to continue, we also look into trying to make this scaffold that we have, the Lantibiotics, to make them active in a gram-negative setting. For this, we have two different approaches that we use. First of all, we had nisin variants that had a tail coupled, of which we knew that they have membrane passing, outer membrane passing capacity, but that part I will not talk about. What I will talk about now is about the destabilizing peptides that we used, and so we figured out to check if we could find synergy between vancomycin and these peptides, or nisin and these peptides. So what we did is we simply looked in the literature and found these peptides, and we selected ones that already had quite a good MIC value by themselves to have a good starting point. And then we started to test synergy. Just to explain the concept, so you make a dose-dependent graph here, and so this is compound A and this is compound B. So if you end up somewhere here, it's not an improvement, it's simply an additive effect. If you end up here, it's very synergistic, and if you end up somewhere here, it's antagonistic. Yeah. So based on where you end up on this plot, it tells you if there's a good synergy or not. So we tested first the peptides and the nisin, for instance, alone. And you should focus, for instance, on these red ones. This is Klebsiella pneumoniae. So you should focus on the 6th and the 8th peptide. You see that's 32, 4, and nisin is 48. Yeah, micromolars it says. But then if we add the compounds, this is not MIC value. So this is a calculation where the value, if it's very low, it's if it's below 0.5, then it's considered synergistic. So what you see is that, especially for Klebsiella pneumoniae, you see that the peptide 6, 7, and 8 show very low Vichy index, which means that they are very synergistic. And you can also plot it in the same way as I showed the figure just now. And we decided to continue with 6 and 8 based on the results that we had. And if you repeat this, so this is nisin. So we decided to synthetically also buy the D8 peptide. So this is an L-form peptide simply, and here we have the D form. And we wanted to do this because, first of all, to see if the target is maybe specific or not, but this is a good indication that the peptide itself is not specifically doing something, but it's more a general thing that the outer membrane is disturbed. What we show here also that D peptides have a slight advantage in terms of stability. So what we saw is that in the end we can find nice results against Klebsiella pneumoniae, but also Acinetobacter baumannii, which really show very low MIC values. So you go very low to 0.5 micromolars, which is, am I seeing this correctly? Sorry, yeah, I think so. So this is very interesting and potentially useful. We also then checked with vancomycin, simply because vancomycin is already applied, and therefore makes the gap to an application smaller. And also with vancomycin we see very nice results. We also now want to try actually to see if we see any effects in gram-positives also, because if you can lower the dose of vancomycin that you have to give a patient, the chances of problems are also lower. Yeah, we're currently doing testing on toxicity and so on. That looks until so far very good, and we're now busy also trying to patent these combinations, and that's ongoing. So what we see is that we can get synergy using these outer membrane permeating perturbing agents. We see low toxicity and hemolytic activity, and we are still doing some more toxicity studies, stability studies, and also checking for immunogenicity. And we're also started collaborations for animal models to test more of the relevant parameters. Also, I want to announce that together with Oscar and Wilfried van der Donk and Jörn Piel and Manolo Montalban, we're organizing a conference in April on RIPS. So that's ribosomally modified and post-translationally, ribosomally synthesized and post-translationally modified peptides, such as nisin. So most of the work that I've shown here was done by Chen and Manolo. Manolo is now at the University of Granada, and Chen is still in the lab. I'm going much faster than I should, but this is it. This is the end.